Okay, so today we're going to look at translocation, and translocation is the movement of sugars and other soluble substances, things like amino acids, etc., what's collectively known as the sap of a plant through the phloem. And what we will see that this is one area where we still don't really know exactly how it works. So we have to evaluate the strength of the image. Okay, so let's have a look in the iBook. So we're based around this area here, finishing off plant transport. Right, so sucrose, because it is a very, very soluble sugar, is the what the plants convert to and transport it around. So this is why we get sucrose from things like sugar cane and sugar beet, etc. Okay, so it takes place in the phloem tissue, and you normally find the phloem sitting next to the xylem, just on the outside of it. So the walls aren't as thick as the xylem, and we will see that they are living cells as well. Let me just find an image of that for you. Right, so this is a longitudinal section, and we can see these are called the sieve tube elements, and we have the companion cells right next to them. So this is just representing what's going on here. So living cells, and we have sort of strings of cytoplasm running up through them, but they don't have a nucleus. So they are connected between them by these things called sieve plates, and we have the companion cells very linked to the sieve plates, to the sieve cells through the plasmodesmata. These are packed with many mitochondria, so these are showing these are very active. We have fibres to give strength, and we have parenchyma, which is just the sort of surrounding packing cells there. Okay, right, so I've covered all of that. Right, so what do we know about how it all works? Well, the first experiment was really done by squirrels. Well, squirrels could do this experiment. So if they do remove the bark from a trunk, then we notice this bulging in the region above it, and sucrose is accumulating there. So what that is telling us is that in this case the direction of the movement is from the top, basically the top of the tree, down to the bottom there. So by removing the phloem, which is just underneath the bark, you're preventing that, so it accumulates there. Some classic examples have been done with aphids, not very pleasant experiments, but you take a feeding aphid, and if you section it like this while it's still alive, then you can actually see the end of the mouth parts are in the sieve tube, so that's proving that the sap is transported within the phloem. Then you remove the head of the aphid, and what you notice is that the sap still drips out from the back of the aphid's head. So this is telling us that the sap in the phloem is under pressure, whereas in the xylem we had it being sucked up. The opposite of that, this is showing it's got high hydrostatic pressure and will leak out anyway on that. Right, back to that in a minute. Right, other classic experiments that we have, what we can see here is if you feed a plant leaf radioactive carbon dioxide, it will convert that into glucose and then be transferred into sucrose and transported around the plant. So what we can see in this case is that it's been transported to the growing point, some has gone down to the roots, and we can see some in there, if you like, in transit. So what we can see from this is we've got actually what's called bidirectional flow, because we've got the radioactivity moving up the plant and moving down the plant, so it's bidirectional. And you can actually do it by combining this with the aphid experiments. You have aphids at different positions. You can measure when the radioactivity appears from the back of the aphid's head. So that you can use that speed equals distance over time to work out the rate of flow that we've got going in there. And this is what we see here, 25 to 100 centimetres per hour. Right, so the main part of this is, the best explanation that we have so far is called the mass flow hypothesis. And let me just show you what mass flow is. So if you take two pieces of selectively permeable membrane and you put a solution of a strong sugar solution in one, a weak sugar solution in the other. And of course this will have the lowest water potential, this will be a higher water potential, getting closer to 0. Water will enter by osmosis, we'll have much more water coming in here than on this side. As a result of this, we will have a build-up of pressure here, and we will end up with a mass flow of the solution in this direction here. And that, hopefully that thought experiment will show that this is possible. So what happens in the plant is that we have the cells in the leaves making sucrose via glucose by photosynthesis, and these then the companion cells actively take up the phloem and they load them into the actively take up the sugars and load them into the phloem. And we call this place where sugars are loaded in, i.e. leaves, sources, and we call places where the sugars are removed from the phloem sinks. So added to in the source, removed at the sink. So this could be taken, say this was a potato, these sugars could be taken by the companion cell out of the phloem, passed into other cells where they be converted to starch for storage. Anyway, hopefully we can see if we're loading at one end, taking away at the other, we'll end up with a concentration gradient of sucrose in there. Because we have a high concentration of sucrose here, water will move in by osmosis. This will call a build-up of hydrostatic pressure that we saw because it oozed out of the back of the aphid's neck, or head rather, and we will end up with the mass flow from source to the sink. And if we are removing from one end and adding at the other, then that pretty much links back to the little experiment that I showed you a moment ago with it going from left to right. Okay, so there are some problems with this. First one, most obvious one, it doesn't explain why these are there. These are going to get in the way of flow, and in biology everything is there for a reason. Bidirectional flow has been observed. I don't think this is an issue because if you've got pressure in the system, it doesn't matter if you open the tap upstairs or downstairs as long as you've got that hydrostatic pressure there. What is a problem is that if you use respiratory poisons, so something like cyanide, on a companion cell down here, so it's not at the source, not at the sink, it will stop this mass flow. So we therefore know it's got to be an active process, which we've suggested from the pumping. However, as I said, it doesn't explain a role for the companion cells that are here. If the mass flow was correct by itself, we would only need companion cells at the source where they can load in the sugars and at the sinks where they can take it out. Right, so what I want you to do today, I want you to annotate a diagram of this showing how sugars are loaded on the source, removed at the sink, build up a hydrostatic pressure and causing the flow down like that. And then it's just the tasks in the iBook here, so 13, 14 and 15. So explain source and sink, and so yeah, written description of this, talk about the evidence, and here's some other ones for you to look up as well, so diffusion and cytoplasmic streaming. See if you can evaluate those as well. Okay, so thank you to those that have uploaded work already. I have provided feedback and I look forward to providing more on this.